All right, so welcome back. This is going to be our last part of our cells lecture. So in this part, we're going to talk about cell signaling, how cells are joined together and interact with each other, and then a little bit about the cell cycle, also known as mitosis. All right, so with that, let's begin. So when we talk about cell signaling, we're essentially talking about how do cells talk with each other? How does one cell communicate with itself? How does the cell communicate with the cell next to it? How does the cell communicate with cells in a different part of the body? All right. So one type of signaling is called autocrine signaling, and you'll talk about these more whenever you go over and study the endocrine system, but we're just going to introduce them now. So autocrine signaling, the term auto is referring to self, right? So this is self-signaling. One cell releases a molecule or some signal that then stimulates itself to then do something, right? So a good example of this are some of the immune cells. Some immune cells will secrete molecules. These molecules can then act on the same cell that secreted it and cause that cell to be stimulated to undergo division and begin replicating itself, okay? Another example, that we have is paracrine signaling, all right? Paracrine, the term para means along the side of, or to the side, or beside. So this is a signal that is going to stimulate the cells next to or adjacent to the cell that released the signal, okay? And so there's an example of this occurring in the pancreas. So there is a cell in the pancreas that releases a product called somatomedin, and this is going to act on the nearby beta cells of the pancreas, and it's going to stop the beta cells and inhibit the beta cells from continuing to produce insulin, right? And so insulin is used in the blood to act on glucose, right, and bringing glucose from the blood. And so when you release this one molecule from one cell, it acts on the nearby cell to have an effect on it, so to stop it from, or to prevent it from releasing insulin. Okay, so autocrine, paracrine, and then you have endocrine, right? And so endocrine is the most kind of probably popular one that we typically think of. Endocrine, instead of secreting signals that act on itself or act on cells nearby, it's going to act on cells that are far away. Okay, and so these are going to be distant cells, like the cells, some of the cells of the pituitary gland will secrete a hormone that then goes and acts on cells of the thyroid, right? So you have a long distance from the cells of the pituitary to the cells of the thyroid gland, and it travels via the blood. Okay, that's the main thing, or one of the key concepts of endocrine system is the signals travel a good distance, and they do so via the blood. All right. So you can also have something called synaptic communication. And we'll talk about synaptic communication in this class when we get to the nervous system and we talk about how one neuron will communicate with another neuron, or how one neuron will communicate with a muscle cell or something like that. But this space here, this kind of region in here with this one cell here called the presynaptic cell, this space in between, and then the cell down here, this whole region is referred to as a synapse. Okay, so it has to do with a neuron communicating with its target cell. All right, so that is a synaptic communication, and it's going to utilize something called a neurotransmitter. Okay, a neurotransmitter, and we'll talk all about neurotransmitters later in future lectures. Okay, now we can also have signaling take place and classify it based on how the signal or how the ligand, and we'll talk about what a ligand means, how it interacts with the cell itself. Okay, so one type is called metabotropic signaling. All right, in this case, the molecule that is initiating the signal is going to bind to a receptor on the outside of the cell. Okay, or this receptor is bound to the membrane. Okay, in other words, it's not inside the cell. Okay, so you have some molecule here. Okay, so say it's insulin or something. It will bind to its receptor, and then on the inside of the cell, you have a sequence of events that take place on the inside. Okay, so the key here is that these signaling molecules are not permeable to the membrane, and so they cannot diffuse freely into the inside of the cell. So they have to bind to the outside, and then initiate a cascade of events on the inside, and then you get some type of response, right? So you increase protein synthesis, or you begin opening up protein channels or something like that, okay? All right, the second type is called ionotropic signaling, okay, or receptor signaling. So in ionotropic signaling, you have an ion, okay? So you have an ion, which here we have our these purple ones here. And this ion is going to bind to a receptor, okay? So it's going to bind to a particular receptor on this protein. When it binds to the receptor now opens and becomes a channel, okay? So now we've opened and we've become a channel, and we can allow ions to flow in here quickly, right? So here's the molecule. It binds to the protein here, which is acting as a receptor. Once it binds to the receptor, now it acts as a channel. It opens up, and it allows these ions to flow into the cell, okay? So this is what we see at the neuromuscular junction that we'll talk about with skeletal muscle contraction. But acetylcholine, which is a type of neurotransmitter, is going to bind to the receptor, so it binds, and then that protein acts as a channel, and it opens, and it allows ions, sodium ions, to rapidly flow into the cell, okay? So this is called ionotropic signaling, okay? So here's an example between the two. This image on the left is showing the ionotropic that we just talked about. It's also called ligand-gated channel. And this image on the right is showing a G protein coupled receptor. This is the example of metabotropic signaling that we just mentioned a second ago, right? So in ionotropic, we're binding an ion, right? Some type of ligand, in this case, a neurotransmitter. We're going to bind it to the receptor. The receptor is going to open, and now it becomes a channel, and it allows these things to flow in very quickly, right? It allows these ions to flow in, okay? In contrast with that, in metabotropic signaling, we bind our ligand, or in this case, a neurotransmitter, to the outside of the cell, so to this extracellular or membrane-bound receptor, and then we initiate a cascade of events on the inside of the cell, okay? So these are things that cannot pass through to the inside of the cell, and you initiate this cascade of events, and in this case, it causes the opening of a channel, and the ions are going to rush in to the inside of the cell, all right? Okay, and then lastly, we have something called nuclear receptor signaling, okay? And nuclear receptor signaling has to do with the binding of a particular molecule. In this case, we're talking about a hormone, a steroid hormone, and it will travel into the cell. It diffuses across the plasma membrane, okay? And in AMP2, you'll talk about how that hormone is able to just cross through. It will bind to a nuclear receptor, so that's what this is. This nuclear receptor can bind to a region of DNA and act as a transcription factor, and that essentially initiates the process of protein synthesis that we talked about earlier, and initiates the production of a particular protein, and then that protein can do something, right? That protein may be an enzyme, it may be a membrane-bound protein that's going to be inserted into the plasma membrane, right? It could be lots of different things, okay? But in this case, we're going into the cell via diffusion, bind to a receptor that acts as a transcription factor to initiate transcription. We create mRNA, and then we then translate that mRNA into a protein using the process that we talked about previously, okay? All right, so now we're going to talk about how different cells interact with each other, okay? So all of the cells in our body are in some way going to be acting independently, so they do their own thing, but they are also acting in conjunction with the cells nearby, right, in some way. So one of the ways they can act with the cells nearby is by forming some type of connection with them, some type of junction, okay? So one type of cell junction is called a hemidesmosome, okay? So this term hemi is meaning partial, so you can probably think that there's another type of junction called a desmosome, because this is a hemidesmosome, there's probably a desmosome, and there is, and we'll talk about that in a second. These help anchor a cell to the basement membrane, okay? So this region down here, this kind of yellow-brown region down here, this is something called the basement membrane. This is where our cell is going to be anchored to. So it helps stabilize it. This region up here, this is a cell, right? This is an epithelial cell. These hemidesmosomes are going to anchor this basal part of this cell into this basement membrane way down here, right? And you have all of these proteins, these integrin proteins, you have collagen proteins, and these are going to form these cross links, and it helps to keep the cells anchored so they don't just easily tear and fall right off. Okay, so there's a hemidesmosome. There's also desmosomes. Instead of anchoring a cell to the basement membrane, these are going to anchor adjacent cells together. Okay, and these are going to use something called cadherin proteins, these green proteins in the middle. They're going to form, you can see one kind of has this concave region, and one is going to kind of have this convex region, and they fit nicely together, and they oppose each other, and they help to keep these cells very opposed to each other. All right, these desmosomes, you know of these desmosomes because if you've ever had a sunburn, what happens to you a few days or a week or so after a sunburn? What happens? Well, you start to peel, right? That burned skin starts to kind of slough off and peel. And when you peel off in kind of little sheets of skin, right? Well, the reason the skin is staying attached is because of these desmosomes. These desmosomes help to anchor the adjacent cells to each other and keep them intact very nicely, okay? All right, we also have one called adherins junctions, okay? Adherins junctions are another type that serve to closely bind adjacent cells to each other, and they're called adherins because they use a protein called a cadherin. Okay, cadherin. That's where we get the term adherins. So these cells are adhered to each other very, very tightly. Okay. Okay. We also have ones called tight junctions. Tight junctions are used to oppose cells extremely close together, so as to prevent things from passing between them. Okay, so passing via this paracellular route, which is between adjacent cells. Okay, it uses molecules called occludin molecules, and so a good example of this are things or are cells that you find in the GI tract. Okay, so think of the cells in the GI tract, in the stomach specifically. We're going to think in the stomach. Well, in the stomach, we also are going to release acid, right? We have hydrochloric acid that we find in the stomach. Well, to prevent that acid from seeping between the cells of the stomach and getting down into the underlying tissue, the cells of the stomach have these tight junctions that oppose the two epithelial cells very closely to one another, and that prevents that acid from leaking between the cells and getting into underlying tissue. Okay, so a tight junction, that's a good example of that is what you find in the stomach. Okay, and then the last type of junction here is a gap junction. All right, so a gap junction, based on its name, you can probably guess what it looks like. It's going to be a gap that connects one cell to another cell. All right, and so where we find these, or one example, is we find these in the heart. All right, and so in the next lecture, we'll talk about histology, and we'll talk about cardiac muscle. And cardiac muscle has these structures called intercalated discs. And as a part of these intercalated discs, they contain gap junctions. Okay. Now, the main purpose of gap junctions is to allow very rapid movement of ions and whatnot from one cell to the next. Okay. Well, if you think about the heart, what does the heart do? Well, the heart's job is to contract, and when it contracts, it squeezes blood out, right? Well, what we want is we want the heart to contract in this nice kind of functional unit, and we don't want it to contract in a very choppy, disorganized manner, right? So the way you allow this, or a way you get this to happen, get this nice kind of synchronized contraction, is you insert gap junctions, okay? The gap junctions allow the electrical signal to flow from one cell to the next cell extremely quickly so that the whole heart kind of functions as a single unit and you don't have this choppy contraction that you would otherwise have, okay? So you have these junctions here. Here they are. This whole protein is called a connexon, and the individual units are called connexins. Okay, so a connexon is made up of connexins. Okay, they can open up, allow things to pass between them, or they can close and prevent the movement of things between them. Okay, all right. So our last little part here that we're going to finish with is talking about the cell cycle. Okay, so the cell cycle is a very important concept in anatomy and physiology because all of our cells, to some degree or another, or most of our cells at least, are going to have some um, ability to be able to reproduce and to make more of itself, right? So, as part of our characteristics of cells and the cell theory that we talked about on one of the very first slides of this lecture, is that all cells arise from previous cells, right? So one cell is going to divide and give rise to the next cell, and then that cell is going to divide and give rise to more cells, and so on and so forth. And so this process here, this is called mitosis, okay? And we'll talk about mitosis in the lab as well. So in general, we have a couple of phases of this cell cycle. One phase is called interphase. The other phase is called mitosis, all right? And then we have something called cytokinesis, all right? So in interphase, we have this phase called G1, S, and G2. We also have a phase called G0 or G0. So in these G phases, we have cells maturing, we have them growing, we have them duplicating their organelles, because the whole purpose of this mitosis that we're going to talk about down here is to take one cell and create two cells, right? And then each of those cells produces two more, so you have four. And then each of those produces two more, so you have eight. And you get this kind of growing of cells, right? So if we're going to make two cells, we have to duplicate all the organelles because they're going to go to two cells, so we need two sets, right? So that's what we have happening in the G1 and G2. We're going to increase the amount of fluid and the amount of cytosol in the cell. In the S phase, the S stands for synthesis, okay? Synthesis of what? The synthesis of DNA, right? Well, why do we need to copy our DNA? For the same reason we need to copy all of our organelles. We're going to make another cell, so we need another copy of DNA to put into that cell, right? So once we finish all of this, we can enter into the mitotic phase, okay? Now, the mitosis phase is broken down into 4 or 5 subphases, okay? Depending on what books you read, it'll have 4 or 5, okay? Primarily, we're going to talk about 4 of them, okay? And these are going to be called prophase, metaphase, anaphase, and telophase, okay? So PMAT. Prophase is going to be characterized by the kind of condensation of the chromatin, and watch one of the previous parts of this lecture if you need to know what chromatin is, but chromatin was that kind of uncondensed DNA. We're going to start to condense our chromatin into nice condensed chromosomes, right? So this is prophase. We're also going to have something called the centrioles begin to go to opposite sides of the cell, okay? And they're going to go to opposite sides of the cell because we're going to pull the cells apart, okay? Or pull the chromosomes apart, excuse me. The second phase is called metaphase, all right? Metaphase has to do with the chromosomes aligning in the middle, okay? So a good way to remember metaphase is metaphase stands for middle, okay? So in the middle of the cell, all of the chromosomes, which are now kind of in that distinctive X shape, are going to align in the middle of the cell at a region called the metaphase plate, okay? And they're going to be attached by something called these mitotic spindles, okay? So mitotic spindles are these fibers that stretch out from those centrioles and attach to the middle of the chromosome. In anaphase, the next part, 4, you can take anaphase A apart. So we're going to begin pulling the chromosomes. We're going to begin pulling them apart, pulling them to opposite poles of the cell. Well, why are we pulling them apart? We're pulling them apart because we're trying to make 2 new cells. And so we're going to pull each copy of one chromosome toward its respective side of the cell. Okay, so that is anaphase, and this is being accomplished by that mitotic spindle and being accomplished by those centrioles. And then we have telophase. Telophase is where the chromosomes arrive at their respective poles of the cell. You begin to have the nuclear membrane begin to reform around the chromosomes. And I did mention in prophase, we begin to have the nuclear membrane kind of begin to break down, which the nuclear membrane is just that nuclear envelope that surrounds the nucleus, right? Because we're trying to get access to our chromosomes and our DNA. And so this happens in telophase. the chromosomes begin to uncondense, so they're leaving the chromosome state and they're beginning to transform back into the chromatin state. And then lastly is cytokinesis. And so cytokinesis is technically not part of mitosis. It happens kind of after mitosis. That's why we have this dotted line here to kind of separate it. And so cytokinesis is referring to the division of the cytoplasm. Okay, so division of the cytoplasm. We're essentially taking this kind of glob of cells that's almost two cells, but it's really one cell, and we're going to essentially pinch it in half. And when we pinch it in half, we get two new cells that are genetically identical to each other, right? So we started with one right here, and it goes through, and we end up with two, okay? And then those new daughter cells can now enter into mitosis again, and they can divide. And then their cells can enter into it and divide again, and so on and so forth, okay? So this idea of mitosis is division of our cells, specifically our somatic cells. So the cells of our body with the exception of our sex cells, so our gametes, okay? That's going to be a phase called meiosis and not mitosis. So this is mitosis, right? And the goal here, the point of this is to make two genetically identical daughter cells, okay? That's what we're trying to go for. Two new daughter cells that are genetically identical to their parent cell, okay? A good way to learn the cell cycle is to look at images of each cell in the various phases and then associate the characteristics of each phase. So, okay, in metaphase, you have the chromosomes aligning in the middle. Anaphase are starting to be pulled apart. Telophase, they arrive at their poles. Cytokinesis, they're being split into two, so on and so forth, right? And so as you're studying mitosis, look at some of the pictures, and you'll primarily need to know what each phase looks like primarily for lab, okay? So for lecture, focus on the descriptive qualities and the descriptive characteristics of each phase of the cell cycle, okay? All right, so that is the end of our cell lecture. I know there's lots of parts and some of the parts were kind of long, so thank you for bearing with me. If you have any questions at all, please email me, please reach out, and I'll be happy to answer, and I'll see you in the next video.